Hello everyone, this is Ketisha. In this video, I'm going to go through the AQA Biology 2025 Paper 1 Higher Tier. Question number 2 is on the topic of organization. Eating too much carbohydrates can cause obesity. Give one other disease that obesity is a risk factor for. That would be type 2 diabetes. Type 2 diabetes. Now you could also say high blood pressure or CHD or coronary heart disease. Cardiovascular disease, you could say stroke or heart attack, arthritis, cancer, sleep apnea, liver disease, or any of them will be fine as well. But the easiest to remember is type 2 diabetes. There are risk of getting it, doesn't mean that they will definitely get it. So table 1 shows information about sugar intake and obesity in different countries. So countries, you've got A to G, and they are at mean sugar intake per person per day in grams, mean obesity rate. Let's look at the lowest one, which is for B. So if I look at B, the mean is only 80, and the mean obesity rate in arbitrary units is only 9. Whereas if I look at the highest, which is 192, which is country G, the mean obesity rate is 32%, which is the highest. So there is a clear link between these amount of sugar people actually intake and the mean obesity rate. Complete figure 2. You should label the y-axis, add the scale for the y-axis, plot the data for countries C, D, E, F, and G, draw a line of best fit. So they've got A and B. So C, right, let's do the labels first. So you've got the mean sugar intake per person in grams. So this should be the mean obesity rate in arbitrary units. So we literally need to copy this. Title for the y-axis, because you've got the mean sugar intake at the bottom. So let me write that. Mean obesity rate in arbitrary units. So the lowest, well, we'll start at 0 because it's going to 9. The highest is 32. So, and B is 9. So this should be 10. And this is 20, 30, 40. So you don't have to label in between. So let's say A was 122. So A is 122. Let me bring a copy of this table so we can actually see this on the side. So hopefully you can see it. That should help. Right. So as you can see, A is 122, which is, so the x-axis, each box is 4. So 22 at 18. So C is 172. 172 should be over here, and 25. So that's 20, 25. So that's C. D is 150, which is between the, so, and 23. 25, 23. That's D, sorry. D is 110, in between 100 and 120, and it'll be 15. That's E. F is 144 at 19. That's over there. That's F. And G is 192, 108, and at 32. So 32 is going to be here. So 190 and 92, that's G. So if I was to draw a line of best fit, what would that line actually look like? Let me get rid of this. Oops. Oh my God. Right. So G is 192 at 32. So 32, that's G, sorry. Okay. G. So it should be a straight line. Okay, so let's have a look at this. We have labeled the y-axis at the scale. We plotted the data and drawn a line of best fit. Right, so, right. Figure 3 shows information about the relationship between sugar intake and tooth decay, which is also similar to what we drew for the amount of sugar intake and obesity rate. Name the type of graph shown in figure 3. Is this a scatter graph? You can also call it a scattergram. What relationship is shown between two variables in figure 3? Inversely proportional would actually look like this. No correlation is where it's going like that. Positive correlation is when the line is actually going up. So it is positive correlation. Sugar and starch are carbohydrates. A student investigated the effect of temperature on the activity of amylase. For each temperature, the student mixed starch and amylase together. Now, starch will be broken down by amylase into glucose. So, starch is broken down by amylase into glucose. Now, you can test starch using iodine. It will go from orange or yellow, orange, brown orange liquid to blue black. And if you want to test for sugar, you can use Benedict solution. Heat it above 60 degrees and it will go from blue to yellow, orange, or red. Right, let's say red. Right, depending on how much sugar there is, it'll turn into more reddish color. So if we mix starch and amylase together, add iodine solution after 5 minutes. After 5 minutes, if there was starch, it will go blue black. If not, it will just simply remain in this orange color. So the table here shows at 10 degrees Celsius, after 5 minutes, it remained blue black. So starch is still there. Starch present. If it goes yellow brown, that means there is no starch. and at 60 and 90, there is still starch. That means the amylase didn't actually work. It only worked at 30 degrees Celsius. Explain the results at 30 degrees, at 90 degrees, right? So at 30 degrees, the starch would have been broken down by the amylase into glucose, and the iodine solution basically remained unchanged. The iodine solution is actually this yellow-brown color. So at 30 degrees, the iodine solution remains yellow-brown because the starch is not present, because the amylase enzyme broke the starch down. But what about 90 degrees? Why is it still blue-black? That's because at 90 degrees, the iodine is changing color due to the starch present. Now, why is the starch present? Because the amylase has been denatured by high temperature. So the iodine solution turns blue-black because starch was still present because the amylase enzyme was denatured. How could the student change the investigation to find a more precise temperature that amylase works best at? How could the student change the investigation to find a more precise temperature? So they tested for temperatures. The test was 10, 30, 60, and 90. So it's not working at 60. There's no point in checking after 60. So you want to check around 30, slightly above or below. So test more temperatures between 10 and 30, test more temperatures between 10 and 60 would actually be better. 10 and 60, that includes 30. Protease enzymes are involved in protein digestion. Different types of protease enzymes are active in different organs of the digestive system. Complete table 3 to identify the organ where each protease enzyme is active. So pepsin is a type of protease enzyme which is present in the stomach. How would I know that it's in the stomach? That's because of the pH is 2. It's highly acidic in the stomach. Trypsin is another protease enzyme, but that works at alkaline solution. So that would be in the small intestine. That brings us to the end of question number 2, and I will see you in question number 3.